appreciate the opportunity to present. Thank you to the organizers. This is a topic near and dear to my heart. I've spent pretty much most of the last 26 years studying Cubozoan envenomations. These present a serious medical urgency and can result in deaths, principally in areas of the Indo-Pacific. Cubozoa comprise over 40 species, and there are two extremely medically relevant families. Shown here are Chirodropid, the larger animal with the 4 corners and up to 15 tentacles per side, Perpedalia, and the Alatina alata, which is a species we have in Hawaii. Shown in the center is a child that survived being stung, and this is in shallow nearshore waters, so it is a serious health burden for folks in principally in the Philippines, but other parts of the Indo-Pacific as well. If you can start the video, please. Sir? Yes. So this is a time-lapse microscopy movie showing what happens to whole, freshly drawn whole human blood. And what one sees here are obviously the red blood cells, but in the center are platelets and then a few white blood cells. So over the course of just a few moments, what you saw was that normal healthy cells, red blood cells, became crenulated, then they swelled, and some became ghosts. At that point, you have loss of hemoglobin or hemolysis. At the same time, the platelets also aggregated and become swollen and ruptured, and the white blood cells became activated. When I started this work, the pet hypothesis in the field was that there was a silver bullet ion channel that was to blame for the deaths. So as a biochemist, I went about to look at the venom, and as we do, using HPLC, I separated according to size, and then I took each of the fractions and looked to see what kind of phenomenology would occur. What we saw, as with the previous movie, was that there was one fraction in particular, the small fraction that you can see about D2, and I don't know if I have a pointer, right after the main first peak, would comprise the rapid death as well as the hemolysis that you just saw in the movie. So I proceeded then to look at a number of different Cubozoa venoms and found the same phenomenology. So I decided to focus in on that and look at the mechanism of action and pathophysiology to see if that could explain these serious injuries, including acute cardiovascular collapse and what is called Irukandji syndrome, which is another life-threatening sequelae. Looking then at just the small 42 kilodalton peak, which I don't have a pointer, so it's frustrating me, that in it of itself was able to recapitulate what I just mentioned to you about the time course to death in a mouse. Sorry. There we go. Okay. Okay, so that peak in and of itself was able to recapitulate the entire phenomenology. So what we did was to treat red blood cells and to do negative staining, and what one sees on the surface is something very similar to streptolysin O and bacterial porins, in that there is a monomer that can self-assemble and form a stable rivet-like pore in the surface of a cell, and in this case a red blood cell, but it is a promiscuous porin and can bind to the surface of numerous cells. When one does the injection of the full venom from various Cubozoa or the isolated porin, I was surprised to see that there is a conserved mechanism of action, and this basically what happens to the animal is you have a loss of ventricular contraction. So these are time series Doppler readings of the heart muscle itself. At the same time, with copper leads, you can measure the electrical conductivity system of the heart. We see that the conductivity system is spared, but the heart muscle stops to contract. This is classical, what's called hyperkalemia, and that is an increase of the plasma potassium. And I won't quiz you on this, but if you remember the Nernst equation, basically if you have a higher level of potassium in the blood plasma, the cardiomyocytes can no longer beat. The good news is that this is reversible. So it isn't a toxin-directed toxicity of the myocardium. It's a transient loss of the ability to beat because of a transient elevation of potassium. And in fact, that hypothesis was borne out with subsequent work, which is beyond the scope of this quick 12-minute overview talk today, but I'm happy to chat with people about more in the details. But what we found then over the course of these 2 decades is that this conserved mechanism of pathophysiology is found again and again amongst various members of the Cubozoa family that we've looked at, both chirodropids and cryptidids. So if one looks basically at this porin binding to membranes, forming these pores, immediately one can have the loss of potassium and then subsequently hemoglobin. At the same time, this can happen in white blood cells to lose histamine and cytokines, and platelets to lose cytokines and catecholamine. So I will show in the next few slides basically a conserved mechanism of pathophysiology that really goes to explain both the acute cardiovascular collapse in various chirodropid stings, as well as the slower, less venom dose Irukandji outcome. But to just take a moment now and to consider what we've found with regard to this porin, it is a monomer that must self-assemble as a ring of pearls, if you will, in order to form this rivet-like pore on the surface of the red blood cell. And interestingly, as I mentioned before, this is very similar to anthralysin O and streptolysin O, bacterial approaches to toxicity. In terms of the kinetic sequence, though, what one has at the beginning, though, before the loss of hemoglobin and the form of a ghost, the ghost cell formation, is the immediate loss of the small ion potassium. And because red blood cells have about 120 millimolar potassium within them, the lysis of a few, just a certain portion of the red blood cells in the body is enough to tip the plasma potassium level over 5 millimolar potassium, which sends you into what's called pulseless electrical activity. Now, what we found is that this is a temperature-dependent phenomenon, and so that if one treats this venom or the purified porin in various conditions, there are different outcomes. So at 37 degrees, you have functional pores being formed rather rapidly, whereas at 45 degrees, 45-minute pre-treatment, one does not see this formation of active pores and very low amounts of hemolysis. That's not the same as complete denaturation, which would be 100 degrees with detergent and SDS. So these data basically show that, and this is published. But interestingly, when you talk about the toxicity of the venom, the HU50, the amount concentration of the venom required to lyse 50% of the red blood cells, you see that it becomes more potent or left shifted at cooler temperatures. And this has been borne out time and time again, which is a big warning I want to give in terms of the first aid management about ice that I will return to later in the talk. Whereas, in contrast, exposure to heat, even skin-safe, rather hot but still tolerable, 42 to 45 degrees centigrade, renders the venom less toxic, and higher doses are required to cause hemolysis. More interestingly, once the full sequence of the porin was determined by Nagai et al., we found that there was a calcium binding domain. So we thought that perhaps one could interrupt this by competitive inhibition using divalent cations. And I went through a whole series of those and found that In fact, copper gluconate at very low concentrations and zinc at higher concentrations could do this in the absence of heat. So this became the basis of a now fully granted US patent, and this product line, this work was all funded by the US military because there were career-ending stings amongst combat divers because of various Cubozoan envenomation. So this has now been in use and is publicly available for 8 years. To review then the mechanism of pathophysiology, if you will, is this fast highway at higher venom doses. So basically, stings by chirodropids with larger mastigophores, larger tentacles, can rapidly result in this amount of porin into the body, which quickly leads you to the pulse's electrical activity and death. Smaller venom doses, even by chirodropids, can lead to catecholamine surge and cytokine storm, which I don't have time to get into the weeds on this, but happy to chat with people in depth if you're interested. And there's more to the story than the porin. These venoms also contain metalloproteinases, lipases, and small molecules. And I'm currently doing constellation pharmacology with Toto Oliveira's lab, and we're looking at the peptides in the small molecule of portions of these venoms. And there, if we look at lumpers versus splitter kind of point of view with regard to the biochemistry, is quite a different story in the small molecules, but they both share this very potent pore-forming protein, the porins. Okay, so kind of going quickly again, in terms of envenomation management, one wants evidence-based management tools. And so in order to do that, of course, models are very important. One of the areas of current controversy in the field is the first aid application of vinegar. So I'm going to discuss that in a minute. But we've also looked at various biochemical based assays to look at hemolytic assays. We've kind of discussed that already, and I've shown you the mouse pathophysiology, but we've looked now at inhibitors and various first aids approaches with regard to ultimately in a piglet, anesthetized piglet time course assay. So just a quick moment, and happy to chat with people further a little more leisure, but vinegar. So published papers have shown that intact tentacles from Scyphozoa, when exposed to vinegar, can have discharge. And folks have extrapolated that to say, well, therefore, the vinegar cannot be a good first aid. The problem with that is that unlike chirodropids, in which, in fact, victims do exit the water with tentacles adherent to the skin, Scyphozoa are delicate tentacles that quickly fall away. And so what's left on the skin are actually undischarged nidae. And so when you do the experiment with undischarged nidae that no longer have efferent, efferent innervation from the tentacle itself and the sensory systems, it's a very different outcome. So with regard to exposure to water after vinegar, there is no rupture, even from Scyphozoa nidae, whereas exposure to saline, which folks are now promulgating as a first aid rather than vinegar, out of the fear that vinegar will cause rupture, these nidae are still competent to sting. And I have to go quickly, so I'm just going to kind of show some of the work, and happy to discuss it later. But basically, it's not over when it's over, so until it's over. So at 1 hour after the treatment, ice looks good in the first hour if you do 45-minute ice pack. But at 15 hours, this red is no treatment. Ice is far worse versus heat. So hot temperature is a far better first aid. So we also have done pig skin assays, applying live whole tentacles from chirodropids, and you can see with the naked eye the damage, and then this technology that's based on the copper gluconate is a game changer. In anesthetized piglets, we've done the same work, applying the tentacles and then looking at various approaches. And so here's your vinegar rinse with a hot water pack, so it's better than the control. Saline with ice is far worse than the control. Vinegar rinse alone is insufficient. Saline is also insufficient and makes things worse versus the copper gluconate technology. So to prevent further venom injection from undischarged nidate firing and to inhibit activity, here's a paper that's published, and happy to chat with you further about that. And just quickly, in terms of the big picture, I do a lot of work in the Philippines, Indonesia, and other places in which we do a collaborative effort. Ask the question, where are these problems occurring? What's the field ecology? And then the biochemistry, and then the evidence-based care. So I see the Jellyfish Blooms Conference as a unique opportunity to gather stakeholders together from all these different areas. Thank you for your attention.